So let's now take a look at the innovations that could change the way we produce our food in the future. A living legend is on the line for us here, Howard-Yana Shapiro. He is known for saving ancient food crops and helping design the crops of the future alike. Over 40 years, he's been studying, teaching, and exploring sustainable agricultural and agroforestry systems. He's worked with indigenous people for this, but also with cutting-edge innovation drivers like X, formerly known as Google X. So Howard-Yana Shapiro, we all look forward to hearing what you have to say and at your presentation.
Thank you very much for this opportunity to be here today. It's an extraordinary meeting. I've been able to participate in parts of it and have enjoyed the presentations, especially the last ones. A friend of mine, Liam Condon, was moderating, and yesterday Matthias Berninger was on, and I was very happy to see what they're talking about and thinking about. So I'm going to speak today for the next 15 minutes about the speed of change. And it's actually, the question is it faster than our ability to respond? And due to some technical glitches, I'm going to have to ask the gentleman who's assisting me to advance the slides today. Next slide, please. So this is an interesting word, and it's used oftentimes to describe issues that are facing us as humanity. Next slide, please. The issues of wicked are big, they're systematic, they're far-reaching. Please click. They have global interdependencies. Next. They're nonlinear. Next. They're economic, they're environmental. Next, please. They're personal, they're shared, they're human. All these words are used to describe some of the most challenging and complex issues of our time, many of which threaten human health. Next slide, please. But there's some things that are happening. I've been in the field for over 50 years. I started farming as a child, working with our family at the age of 5. And now I look at things like computing and analysis and genetics that enable us to reconsider what biology is. And when I say the word biology, I mean in the broadest sense. Biology as in systems biology and soil science and the microbial community. So all of these things, including plant genetics and how we think about plants that we want to grow in the future, these are the three things that have come together which are changing our lives. Next slide, please. So imagine a chip that has 1.2 trillion transistors on it. It's unimaginable in my lifetime that we have chips like this, but we do. And so the ability to look at things and make analyses is greater than it's ever been. And to me, this is the difference between the Wright brothers flying the first airplanes and going into space. Next slide, please. And we can analyze things at the molecular level in a way that we've never been able to do before. And the cost of doing this is so modest anymore that everyone has this as a tool available to them through mass spec. Next slide. And genetic technologies are more accessible as well because the cost for doing this work is become so de minimis and so modest at this point. And I hold in my hand, let me just grab it right here, what is the future of sequencing. This is an Oxford Nanopore sequencer. You open it up, put the DNA in the carrier, close it up, plug it into the USB port into your computer, and you can sequence DNA with this little tool versus the original sequencers that were the size of a microbus. Next slide, please. But food production is largely considered the biggest threat to our planet. Next, please. Jerry Nelson wrote in the Washington Post a couple years ago that we must shift our emphasis from food security to nutrition security. And this is a quantum leap because it's no longer just the calories that can be produced, it's the complex nutrition. And it's not the nutrition of our parents who thought about a pyramid, so many vegetables, so many grains, so many fruits per week or per day. It's a totally different understanding. It really is the evolution in food and nutrition that we all wish for, but now it's happening. Next slide, please. And global food production by the numbers. Next, please. Biodiversity loss. Next. Freshwater use. Next. Greenhouse gas emissions. Marine stocks depleted. Most chemical uses. And then loss of topsoil. What if we could reduce by 50 or 60 or 75% all of these categories? Then global food production is not the most destructive part of our society. If we could have plants that use 65% less water or 75% less water, or crops that made their own nitrogen, or we could grow twice as much on half the land. These are the things that are really driving and must be understood for innovation for the future. Next. And you hear people say, we have no idea how to feed the planet without frying it. So we must get better at growing more on less land. But all of this is available to us if we accept and utilize the best technologies to us. Next. And we all know that we must fix food. And if I walk down the street and talk to 10 people, every one of them is an expert on foods. But as Jason Clay wrote, no single actor, not me, no publication, not Nature, will make the global food system more sustainable. And this also includes the companies that supply the raw materials that go to the factories for the material that comes to our grocery store, sometimes processed, sometimes less processed. No one of those groups can actually make the change. Next. And then we see things like this. Shortage of food, or the spike in food prices, the index on the left. You see all of these things happen, and how much political instability is caused by food prices and food riots caused by scarcity. Next slide, please. And if you study that little seed that's in the palm of my hand, that seed weighs 1/600th of a gram. And from that 1/600th of a gram grows a tree for 2,000 years that weighs as much as 6 747 airplanes, and it's called a giant redwood. We're having trouble with video, so I can't show it to you today, but trust when I say that in that seed is encapsulated enough information to grow a tree for 2,000 years. Next slide, please. And look at the genius of humanity. Modern corn on the right-hand side, Teosinte mexicano on the left-hand side. In the middle is a hybrid between the two. Imagine starting with a plant called Teosinte and ending up with modern corn by making selections over 7,000 years. We don't have that kind of time frame any longer to make this. We need to have quick solutions. We need to have resilience in the systems, which is the ability to respond quickly to a problem. Next slide, please. And this is from 1931 in the state of Iowa. This man had just won the grand prize at the Iowa State Fair. His maize is amazing, but it pales in comparison to what we have today. And much of what we think about is sort of based on these sorts of metaphors. And there's a divide in the world. It's a politic, if you will, between traditional plant breeding like this and modern plant breeding, which will solve many of our problems. And somehow we need to remove or ameliorate the complexities of that negative conversation about modern and old. Next slide, please. And as a young man working on my first degree, I would walk through fields like this and make decisions with a piece of paper, taking notes. This plant looks good, put a ribbon on it. So I would save the seed from that. But now with the type of chips we have, with the sequencers that we have, I can look all the way down to DNA, which has changed the way we consider even a plant and all of its methods and all of the ways we think about it. Next. So these are my heroines and heroes that have moved the world for me as a plant scientist, as a geneticist, as a molecular biologist. Next slide, please. First is Barbara McClintock, the great cytogeneticist who worked on traditional mazes, colored mazes. Next slide, please. Her contribution and the winning of the Nobel Prize was based on transposons. She identified that the color kernels could move between the plants through pollen, which was done naturally, but she inferred that we could do this intentionally. So the ability to move from one site to another site in the cell's DNA really was a breakthrough, because now we understood how we could deal with the complexities of plants. I want this characteristic and this characteristics. We can make the crosses that will give it to us. Next slide, please. The great Gerd of Kusch, who shared an office space with me for many years at UC Davis. He did one thing after having made it clear that human hunger and desires are elastic, but land is not. How do I produce the most productive rice plant in the world? When he was at the International Rice Research Institute. Next slide, please. And this is what it took, this diagram, to produce IR64, the highest quality, high-yielding mega variety that's eaten by more people still in the world today. All of the boxes with green lettering are all the parent lines that were necessary to make the crosses that ended up at the bottom of the diagram in IR64. Years and years and years of intense scrutiny of activity. Next slide, please. The great Norman Borlaug, who I was fortunate to associate with in Mexico, makes a very great statement. Man seems to insist on ignoring the lessons available from history. Next slide. He and the colleague M.S. Swaminathan from India introduced a program that was become known as the Green Revolution. It helped Mexico, India, and Pakistan avoid incredible problems from the wheat falling over and then not being able to be harvested because it lodged in rain and wind. So it was a very tall wheat. And so they developed a dwarf wheat that wouldn't fall over. Now, the Green Revolution was a solution to a problem in a certain moment. No one thought that humans would be so lazy as not to improve it and improve it and improve it. So there's criticism of the Green Revolution by the same people who never worked to improve it. Next slide, please. And Frances Arnold, a chemical engineer. Why is she in here? Because we're discovering that nature can do chemistry we never dreamed possible. Next slide, please. And so she's worked on directed evolution of enzymes. So what could enzymes do for agriculture, do for food? Well, they could replace toxic chemicals in agriculture. What else could they do? They could help work the soil to make the soil more respondent to sequencing carbon or any other number of things that are necessary for healthy plant growth. So the idea that we can create enzymes that we have never had access to before is incredible as another tool in our toolbox to move towards a resilient food system. Next slide, please. And down the road from where I live at the University of California, Davis, is Jennifer Doudna at the University of California, Berkeley, who recently won the Nobel Prize for her work on CRISPR with Emmanuel Charpentier. The next slide, please. The power to control our species' genetic future is awesome and terrifying. So we have this tool now called gene editing and CRISPR, these tools, which allow us to do things that we never thought we could do before. End critical pathogens in plants where they have no resistance, like the banana and the Panama race 2 disease, which is wiping out plantains, which are a food banana, and the Cavendish, which is the dessert banana. But we never had this opportunity before. And there's debate, whether you're in Europe or the United States or China or Africa, South America, Asia. The debate is misguided in many ways. This is only a tool. How we use it is up to us. If we revolutionize the food system using CRISPR, which is a democratized technology, then we really can change many of the problems that are facing us in the food system through innovation. And the last slide, please. This is not a famous statement written by a great agriculturist. It's by a guy who writes about motorcycles. No matter how many expert opinions you line up, you can't vote nonsense into truth. And why I say this, there have been moments in time when experts lined up and said, no, you don't do that. And then people worked steadily on these problems, and the information changed, because what people thought was truth was actually nonsense. So I want everyone to think, when someone says, don't do that, don't think about that, that's an impossibility. Tell them, you cannot vote nonsense into truth. Thank you.
Howard Shapiro, thank you so much for that presentation. And let me just pick up from it straight away. I'd just like to ask you, so you didn't stop at where the limits are. Where's the limit?
Well, I don't know that I know the limits. I don't have a crystal ball. What I do know is that I have to live in a technological world and take the best of those applications and move them forward. I've suffered from heart disease from a young person. If I didn't advance technology in treating cardiological issues, I would have died long ago. But I've been riding a wave of breakthroughs. And I feel the same way about agriculture. I started in the field, walking the fields. We started to understand what would be done with the sequencer. But we always knew that it started with 2 parent lines in a crop. This is what Mendel discovered in his garden. So now when these tools are made available to us, we certainly have to have a global dialogue about their utilization. But much of what we think sometimes is, oh, that's scary. We've done much more than that if you look at teosinte to modern maize in that transition. So my urging is that understand the technologies, understand the innovation, understand what these problems, how they can be solved with these technologies, and embrace them carefully, embrace them with good ethics behind them, but embrace them.
Well, you mentioned speed, that the speed of change is faster, I'd say, than many of us can actually process it. And it's one thing to have tomatoes ripening on your shelf for 6 months. The other thing, the next step is probably those cows without horns. But when there's some kind of interference in what actually is our everyday reality, not to mention human genetics, that's what really when a line is crossed. So where do you draw that line for yourself?
Well, I don't know that I am in a position to make a call on all of that you just inferred. But if you've ever been around a cattle ranch and when they dehorn the cows, it's a horrible process. So if you could have a cow without a horn, it seems like it saves the animal welfare, in particular of that circumstance. If we could have crops that use 65 to 75% less water, wouldn't that be a benefit to many parts of the world that are suffering drought and irregular rains? What if our crops could make nitrogen? In 2018, I published a paper with colleagues about the first time we've ever seen a maize that actually made its own nitrogen, pulling it atmospherically. So this is not scary. But in order to get it into a modern crop, will probably take engineering. But what if all cereals and grains could produce their own nitrogen? maybe not all of it, maybe only 50% of it. That reduces the pollution and the energy to produce those products. What if we can make plants that are resistant to pests and diseases? It eliminates an entire range of pesticides. This is what we have to do, because not everyone lives in the global north, where you go to the ag supply store and buy whatever you need to apply to your land. Many people are working on marginal soils, with marginal equipment, with marginal genetics. So that has to change as well. Otherwise, the only thing we can do is cut down the rainforest to expand the footprint of agriculture, which is something that no one wants to see happen.
How can you be sure that not both will happen? Is that kind of level of innovation that you just presented letting policymakers off the hook?
I couldn't hear you the last part.
Is that level of innovation, that speed of innovation techniques, in the end, not just also letting policymakers off the hook?
No, if anything, it'll make policymakers heroes, because the women and men who make policy, who embrace technology, and again, some of it is scary to the average consumer. You say the word edit genes or CRISPR, and you explain what CRISPR can do, and you show a diagram of making a SNP and replacing it in the DNA. But it's not scary, and we have to change the conversation to the negatives and portray the positives for the betterment of humanity. When you get to human engineering, it's a very complex subject, but there are certain diseases that will never be conquered without the ability to edit. And we have to be careful about it, and we have to be moral about it. But to hold it back to save people from suffering, whether it's through chronic hunger and malnutrition or disease, is, I think, unethical and immoral.
Let's just hope we don't get lost in that wide open space of innovation and possibilities that you've mapped out for us there. Howard-Yana, Shapiro, thank you so much for sharing your expertise and your insights, and also giving us a bit of a glimpse of what you believe is possible and should be. Thanks so much.
Thank you so much.